Um, sure, I could start with that. I think the variants are a wild card.
Mm-hmm.
You know, with the rat study, the exposures are actually, I think, more intimate than occurs in the general population. So I think there is a high likelihood of transmission that we see in the rat study that we would have to account for in a model. Um, but the variants, who knows? You know.
Yeah, who knows is the right answer, but I figured you guys might know better.
Well, I would say that your speculation is probably right on the money as far as our best estimate goes, because it is certainly true that the relationship that I talked about between the antibody levels and the probability of infection is, although it is plausibly causal, it is also plausibly phenomenological, meaning that, and in fact, there's some data to indicate that it may be phenomenological, meaning that like the level that you drop your antibodies to is also corresponding to the amount of evolution that the viruses actually are experiencing ongoing. There's evidence that SARS-CoV-2 is not any different from the human cold, the HCOV viruses in terms of all these variants that we've seen. Like, we followed them in detail in SARS-CoV-2 because this has become a national obsession because of the enormous impact it has. But the common cold varies with genetic change over time as well, in very similar ways, in the very similar way on the spike protein. So there's a lot of evidence that these are very, very similar viruses in terms of just their overall way of making their presence known to us, so to speak. So I think that that's about right. I mean, it may be a little bit longer, it may be a little bit shorter. One caveat that I would say to that, because the estimate is 1.5 years or so, there's also some very weak evidence, because again, in a pandemic, it's really hard to know regarding the seasonality of this virus. It seems very seasonal. We've got a lot of sort of ancillary evidence saying it is, but we can't really nail that down when we've got a pandemic with lots of interventions going on. So all of that comes together to say, it seems like, I would say probably around a yearly vaccination booster would be about right. Now, there's one caveat that I just want to say, which is that the vaccines really are not the same thing as experiencing a natural infection in a variety of different ways. But the one maybe possibly hopeful note is that in order to generate these mRNA vaccines, for instance, they actually have to stabilize that spike protein in certain ways. Mm-hmm. Now, it's not clear whether that stabilization is something that has an effect on how your immune system actually recognizes the spike protein or not. But from what we've seen so far, it seems to help that recognition process. And it may be that having a more stabilized spike protein means that you get antibodies that are going to have a longer duration of immunity against variants as they evolve than normally would be seen. On the other hand, we've already seen some degradation of the immunity in some of the variants that are coming now. And a lot of that degradation is not selected degradation. It's just coming from random changes because it's emerging in places where there wasn't necessarily a lot of vaccination.
Yeah, it's kind of a volatile situation. One more follow-up question. Does anyone else have a question? I see something in the chat. Oh, Dr. Zeiss has a question from Dr. Townsend.
Yeah, just trying to figure out how to answer it. The technology, I mean. Actually, I could just read the question. That might be simpler.
Mm-hmm.
Could you elaborate on why the virus that we used for vaccination represents a better model for vaccination than would be natural infection by the first virus exposed the rats to? So the analogy is using cowpox to vaccinate for smallpox. So they're similar viruses, and there's cross-protective immunity. The RCV, the immunity that it gives is slightly less than if you were to give the natural virus. This is not identical to vaccination. So we don't have a vaccine for SDAV. Such a thing does not exist. And we couldn't really use the vaccine for SARS-2 because the viruses are not similar enough. So this was essentially the closest thing that we could get to a vaccine. And I think the results would be very similar to giving the natural infection to SDAV. They would be very similar, although RCV has got a slightly less, it's going to have slightly less protection.
Very interesting. Did you have a follow-up question, Dr. Townsend, or?
No, I understand. Thank you.
Thank you. So I'd like to ask a question, another one. So when they first came to us, NSF, and said, we'd like you to do this rapid on a COVID information commons, I actually started Googling, coronavirus and looking for background and stuff. And at the time I found an NSF award, which I can't find anymore, which was for the 3rd biennial coronavirus workshop in 1985. And then I went a little deeper and I found some documentation from Springer, and I just put it into the chat. So there's coronavirus history information from Springer and Nature. We all abide by them. We all play with them. I publish with them. A lot of us probably do. And interestingly, if you go through it, you can find papers from the 1960s, right? And we know that the coronaviruses were first discovered back then, I think by a woman in the UK, perhaps. So what is it we think we may have learned if we actually paid attention all those years? Is there something that maybe would help us looking back? Or do you think we just have to look forward?
Well, I have one maybe anecdotal thing to say, which is that one of the more interesting papers that I read was a paper by Callow, which I can't remember the date of, but it goes way, way, way back. And that paper did do some they did some challenge experiments, which are unusual and not often done. But they had some interesting results about this question about the variance versus the antibody decline. And the interesting result was that there were instances where they did, and I can't recall exactly how they did the perfect controlled test, but I remember reading it and believing it was pretty well controlled, where they looked at whether or not if you took took someone who was exposed to a virus and then 2 years later exposed them to exactly the same virus, and whether you took them, exposed them to a virus and then took a virus that was circulating 2 years later and exposed it to them to see which one would infect the person. And only the one that circulated for 2 years infected the person at all. They could not get the old virus to which that person had been exposed 2 years ago to infect the person and cause symptoms. So it just goes to show you that I think that goes along with my sort of anecdotal argument that the generation of these new variants, the evolution of the viruses themselves, is a lot of what we're dealing with in terms of the waning of antibodies, and not so much just a too low antibody level. Nonetheless, I don't think that contradicts the kind of results we can get using those antibodies as a metric for how long you were exposed to that. It's certainly true that having more antibodies is going to help you in defense against a pathogen that's attacking you. So, so the two are mixed in an interesting way that needs further work.
I agree. So I think that would be an interesting paper. I was actually hoping that with the Kick Student Paper Challenge, one of the students would do that, but I think it would be interesting to look back and see what we may learn from the past, other research that's been done while we're trying to plan the future. Any other comments or questions today? Well, thank you all for being here. Thank you for the great presentations. Thank you for sticking with us. In our next meeting, actually, we are going to have one of our Kick Student Paper Challenge winners presenting, which is really exciting. And so we'll be posting those papers on the Kick website, and then we're also going to be adding them to the Columbia Academic Commons. So for the undergraduates that actually participated in one. So we're very excited about that. So we hope you can join us. And Helen, anything else?
No, thank you again to everyone, all the speakers today and everyone who's joining us. And we hope you continue to stay engaged with the Kick.
Thank you, everyone. Be well.
Great.
Stay safe. Thank you. Bye-bye.
Have a great evening.